So now that we've discussed our adaptive immune system's cell-mediated response, it's time for us to start looking into the humoral response, which is the other branch of our adaptive immune system. The humoral response is also known as our antibody-mediated response, and this response is facilitated by our B cells. B cells are the drivers of our humoral immunity, just as our T cells are the drivers of our cell-mediated immunity. Activation of B cells happens in just about the same way as activation of T cells. There are some differences, but the overall process is relatively the same. B cells will have what's known as the B cell receptor, and instead of requiring antigen presentation, B cells will be able to activate themselves based on binding to some type of pathogen with their B cell receptor. Helper T cells can also aid in the stimulation of B cells through the process of co-stimulation, which we saw with the cell-mediated immunity also. Once B cells have been stimulated and activated by helper T cells and other pathogens, they will also undergo the process of clonal selection, the proliferation and differentiation we saw with the cell-mediated immunity. Only B cells are not going to produce clones of themselves. They're going to produce another type of cell known as plasma cells. The major role of plasma cells is to produce and release antibodies, and they do so much more efficiently than our B cells do. We'll also be producing memory versions of our B cells also. Once again, just as our memory T cells, memory B cells will exist for decades and be prepared in the event that antigen makes its way back into the body. So our humoral immune response can be classified as both primary and secondary. And I would actually prefer to call these the first and second humoral responses because I think that would be a little bit more accurate. The primary humoral response simply is the first exposure to a particular antigen. This slide shows us the primary humoral response. When some type of antigen is recognized, that B cell is going to see that and become activated. And the helper T cell could also be another factor that would stimulate and activate a B cell. Once our B cells are activated, they're going to undergo clonal selection and form many plasma and memory cells. Once the infection is subdued, the plasma cells will eventually waste away, but the memory B cells will remain for longer periods of time to make sure that we can deal with that pathogen later on in life. The two products of B cell clonal selection are the plasma cells and the memory cells, and each one of these cells has very unique characteristics. Plasma cells' main roles are to produce antibodies, and they do so much more efficiently than our B cells do. You can see that plasma cells release up to 2,000 antibodies per second. That is a lot of antibodies. Plasma cells will stick around for a few days, however they will eventually break down. Our memory cells, on the other hand, are going to remain. After the initial contact with the antigen that initially stimulated our B cells, the memory cells will remain in case that antigen makes its way into our body later on in life. When this happens, our memory cells will eventually come in contact with that substance and undergo the process of clonal selection once more. The only difference here is that the secondary humoral immune response is much faster and much more powerful than the primary humoral immune response. We're going to break down the most important components of the plasma cells and the memory cells here next. We'll first start with the antibodies that come from the plasma cells. As I described at the beginning of this presentation, antibodies are Y-shaped proteins. We can see this rough Y shape here. There's two heavy chains and two light chains. These large central ones are the heavy chains, and the smaller ones over here are the light chains. Each antibody has a constant region and a variable region. The constant region does not chain, and that can be seen over here. But the variable region can change based on the antigen that our body is fighting off. There's two antigen binding sites up here at the variable region, so each antibody can bind to two different antigens. Another word for antibodies are immunoglobulins, and we often abbreviate them as IgGs. There are 5 classes of antibodies: IgMs, IgAs, IgDs, IgGs, and IgEs. Each of these antibodies are secreted by our B cells in response to specific antigens. Certain antigens will stimulate different types of antibodies, and I'm not going to stress all of the different functions of every different type of antibody, but I do want you to understand what 5 types of antibodies that we have. The major role of antibodies is to go out and bind to a particular antigen. Once the antibody binds to the antigen, it forms what's known as the antigen-antibody complex. And from here, this is going to signal to our immune system that the structure that is bound to the antibody needs to be destroyed. There are four major mechanisms of antibody function. They are neutralization, agglutination, precipitation, and complement fixation. And we can see all four of those antibody mechanisms shown here. One of these that we're not a stranger to is agglutination. We saw this with blood typing when we infuse the wrong type of blood into a patient. Agglutination refers to the clumping up of cells. Since each antibody has two binding sites, one cell can be bound to one binding site and one cell can be bound to the other. Precipitation, on the other hand, is just like agglutination, however there's much more cross-linking. When masses of particles are continuously linked together, they'll eventually get so big that they'll eventually settle out of the solution and become a solid, which we call a precipitate, and that is why we call this precipitation. Complement fixation is another one of our antibody mechanisms, which we saw when discussing the complement system. Antibodies can bind to foreign cells and signal the complement proteins to come in and form a membrane attack complex. This in turn will form a pore in the membrane and eventually lead to cell lysis. Our last mechanism of action of antibodies is neutralization, and this is very similar to the process of opsonization that we saw when discussing the complement proteins also. Neutralization is when our antibodies will bind to the outside of the cell surface, covering up all of the dangerous receptors. Neutralization can also work on toxins that are secreted by these various pathogens. By taking antibodies and binding them to these substances, that can prevent the attachment of those substances to our own cells, therefore neutralizing the danger posed by these substances. So we can thank our plasma cells for producing as many antibodies as they do. Plasma cells truly are antibody-producing machines. And while B cells can produce some antibodies, plasma cells do it much, much more efficiently. We also wanted to elaborate on our memory B cells, and that's where we're going to see them coming and playing their role with the secondary humoral response. The secondary humoral response can be looked at as simply the second exposure to a particular pathogen. The primary response was the priming of the adaptive immune system. We have to expose our B cells to the pathogen so that they can produce the other cells that are going to be very beneficial for fending it off. Oftentimes, this takes time. But what the humoral immunity lacks in the primary response, it makes up for in the secondary response. You can see that this graph here is comparing the primary humoral response with the secondary humoral response. At this moment, we have our first exposure to whatever the pathogen is. And while there is an increase in the amount of antibodies in the bloodstream, that increase is not drastic. We see on the y-axis we're looking at the antibody titer, which is just a way for us to measure how many antibodies are in our blood. The first exposure does not give us a very impressive amount of antibodies in the blood. However, after the second exposure, you can see the concentration of antibodies is going to skyrocket. This in turn makes our secondary humoral response much more effective and much more deadly to the substances that are making their way into the body. One thing that you'll also notice about the secondary response is that this increase in concentration is much, much quicker than the first exposure was. So while the adaptive immune system is a little bit slower to get started, it's much more effective during that secondary response. This secondary humoral response is truly what we mean when we say memory. Now that we can see a little bit about how our humoral immunity works, we're going to separate our humoral immunity into the active humoral immunity and the passive humoral immunity. Both active and passive humoral immunity can be naturally acquired and artificially acquired. But whether or not the humoral immunity is active or passive is what's going to set the stage for how this branch of the immune system is going to help out. Let's first describe the difference between active humoral immunity and passive humoral immunity. Our active humoral immunity is when our B cells are actually encountering antigens and are working hard to fend them off. Just as we saw with B cell activation, active humoral immunity stimulates our B cells into clonal selection. This leads to plasma cells and, more importantly, memory cells. With active humoral immunity, we're going to see memory. We will not see that with passive humoral immunity. So let's jump to this slide real quick to explain the difference between passive and active, and then we'll talk about how each of these can be naturally acquired and artificially acquired. While active humoral immunity refers to the challenging of B cells and the production of antibodies, passive humoral immunity is a little bit different. Passive humoral immunity skips the actual challenging of the B cells and simply administers antibodies into the patient. Since our B cells are not challenged with passive humoral immunity, they will not provide memory in these instances. So to distinguish between active and passive humoral immunity, we need to understand that the source of the antibodies is what's really making the difference. With active humoral immunity, our B cells are fending off the pathogens and therefore producing the antibodies and making memory cells. With passive humoral immunity, we administer antibodies to the patient, and that does not leave the B cells challenged, so therefore no memory is produced. Now both of these can be either naturally acquired or artificially acquired. Naturally acquired is basically what we've been speaking about this entire presentation. With different infections and different pathogens that are trying to make their way into our body, we can naturally acquire active humoral immunity when we're exposed to any type of pathogen. Naturally, throughout the course of our lives, we're experiencing and being exposed to many different things. Our B cells are encountering those substances and producing antibodies to fend off the antigens that are coming from them. We call this naturally acquired because this is a very normal process that happens with an intact immune system. However, the active humoral immunity can also be artificially acquired as well. We can prime the immune system by artificially administering vaccines to our patients to allow them to encounter weakened versions of the pathogens. Vaccines give us the best of both worlds. As we're not administering full-fledged pathogens, we typically attenuate those pathogens to make them less harmful. So weakened pathogens aren't going to be able to cause as much problem inside of our body. So we typically do not have as severe symptoms as we would during naturally acquired infections. But at the same time, this will establish immunological memory to the substances that we're artificially administering. Many very dangerous bugs have either been eradicated or nearly eradicated thanks to the use of vaccines. So while our B cells are still challenged with artificially acquired active humoral immunity, since we are giving the vaccine of our own accord, we classify this as artificially acquired. So active humoral immunity in sum is when our B cells are encountering an antigen and eventually producing the antibodies and in turn producing memory cells. Passive humoral immunity does not challenge the B cells and therefore does not promote immunological memory. Essentially, the passive immunity is when we're administering antibodies from another source entirely. Naturally acquired passive humoral immunity is what occurs when the fetus obtains antibodies from the mother. This is a very natural way to get antibodies from one individual to the next, and this happens normally through development. Artificial passive humoral immunity includes different immune serums, such as an antivenom after a snake bite. Artificial passive humoral immunity can be used when our adaptive immune system is not going to kick in quick enough to help us to survive. Oftentimes, snake venom is deadly enough that we will not live long enough to be able to give that second response. So by administering the antibodies artificially, we can save the patient's life. Now we must remember that passive humoral immunity does not challenge our B cells and therefore does not allow them to stimulate memory B cells. We're simply giving the antibodies, and as soon as the antibodies degrade, we will lose the immunity that we acquired from these methods. So in review, the humoral immunity can be either passive or active. Active humoral immunity is when our B cells encounter an antigen and produce memory cells for that. That can be either naturally acquired or artificially acquired. Passive humoral immunity is when we have to get our antibodies from somewhere else because we're either unable to make those antibodies altogether or not able to make those antibodies fast enough. I've posted a few summary slides here to discuss a little bit about your antigen-presenting cells and your lymphocytes. There are 3 slides in particular, so make sure to look over these so that you have a good idea of what each of these cells do. I post these summary slides so that you have a very quick and easy way to get information about each of these types of cells. So make sure that you take some time to look through these summary slides and that you're familiar with the different APCs and lymphocytes that we have in the body. To finish up the immune system, we have a few disorders that are related to the immune system that we're going to discuss. The first of which is AIDS, which stands for Acquired Immunodeficiency Syndrome. AIDS is a condition in which people experience lots of different infections due to the progressive destruction of the cells of our immune system. This condition is caused by the human immunodeficiency virus, or HIV. HIV is often transmitted in body fluids where it normally resides. HIV affects patients as it directly attacks the patient's helper T cells. As we remember, helper T cells are the coordinators of the immune response. So if those mediators are being attacked and eliminated from the body, we're not able to mount an effective immune response. Oftentimes with AIDS, infections that were once seemingly harmless seem to overwhelm the immune system. Transplant rejection is another disorder that we see related to the immune system. There are different types of transplants, and if not carried out properly, our immune system starts to recognize that the transplanted organ is foreign, and in response to this, our own immune system starts to reject that organ. The 4 types of transplants are autografts, isografts, allografts, and xenografts. Let's discuss autografts first. Autografts are typically the most successful type of transplant because it refers to taking tissue from one location and moving it to another location from the same person. An example of an autograft would be taking a blood vessel from the lower leg and moving it to some other place in the body. These are generally successful procedures as autografts do not contain any foreign tissue. Isografts are another type of transplant that are also generally successful, as isografts refer to transplants that are taken from a genetically identical person. This would be an identical twin. While autografts and isografts are the most successful types of transplants, allografts are the most common types of transplants. Allografts are when tissue is taken from one person and transplanted into another person that is unrelated. Oftentimes, the transplant that is needed is for an organ that is failing in the person, so we don't have an extra one sitting around. Lastly, a xenograft is a transplant that is taken from one species and put into another. These are generally unsuccessful type transplantations, but there's a lot of research going into this area. One of the vital elements to successful transplantation are matching up the MHC proteins. Remember, the major histocompatibility complex is one of the most important self-antigens. So if we start to see different MHC proteins in a particular patient, that's going to start to raise a lot of questions for the immune system of the person who received the transplant. If the immune system is able to recognize the transplanted tissue as foreign, it will often start to reject that tissue and attack it. For this reason, it's very important to match up the MHC proteins of the donor and the recipient. But along with this, we'll need to put patients on immunosuppressive drugs to keep our immune system from recognizing the foreign tissue. One of the downsides to this is that patients on immunosuppressive therapy will not have as strong of an immune system. One of the very important disorders that we're going to discuss in relation to the immune system are autoimmune disorders. Autoimmune diseases is when our own immune system attacks our own tissue. A few lecture videos ago, I discussed the concept of immunocompetence. In order for a cell to be immunocompetent, it first needs to be able to recognize foreign substances and destroy them, but it also needs to be able to recognize self-substances and not attack those antigens. So if there are cells that are not programmed properly, or perhaps self-reactive cells have evaded our own immune system checkpoints, this can give rise to different types of autoimmune conditions. We've seen some of these autoimmune conditions throughout the course of anatomy and physiology. One we've seen so far in this class specifically was type 1 diabetes mellitus. Type 1 diabetes is an autoimmune condition where our own immune system is attacking the beta pancreatic cells inside of the islets of Langerhans. These are the cells that produce insulin. So if these cells are being attacked constantly and eventually destroyed by our immune system, we're not going to be able to produce insulin. So this is an example of an autoimmune disease that affects the pancreas. Another one that we saw in this class was Graves' disease. We saw the thyroid gland was affected by Graves' disease. Graves' disease results in an increase in basal metabolic rate and increased activity of the thyroid gland. A couple of these conditions were specific to Anatomy and Physiology 1 as well. Rheumatoid arthritis is an autoimmune condition that affects the joints. The tissues and synovial linings of our joints are viewed as foreign substances, so therefore our own immune system starts to break down our joints. This can lead to lots of pain and the breakdown of the tissues that are cushioning the joint. We also saw multiple sclerosis when discussing the nervous system. Multiple sclerosis is an autoimmune condition that affects our myelin sheaths. Myelin sheaths in the peripheral nervous system are formed by Schwann cells wrapping around the axons of our neurons. When the Schwann cells are destroyed, we lose the myelination around our neurons, and that significantly decreases transmission speed. Myasthenia gravis is another condition that you likely heard about in Anatomy and Physiology 1. This condition will specifically affect the neuromuscular junction. So this autoimmune condition affects the transmission of signals from one body system to another. Glomerulonephritis is one that you likely have not heard about up until this point. But judging by the name of this condition, nephro means kidney, and glomerulo refers to the glomerulus, which is the capillary beds that can be found in the kidneys. We'll discuss these components a little bit more when we get to the urinary system. However, using our understanding of anatomical terminology, we can deduce that this autoimmune condition would specifically affect the kidneys. And we also have systemic lupus erythematosus. And the word systemic is going to indicate that this condition affects many different body systems. The kidneys, the heart, the lungs, and the skin are all affected by systemic lupus erythematosus. So there are many different types of autoimmune conditions. Some of these we've seen, some of these we haven't. But all autoimmune diseases share the fact that our own immune system is viewing our own tissue as a foreign entity, and therefore our own immune system attacks ourselves. That's why we call it an autoimmune condition. The next disorder of our immune system refers to hypersensitivities. These are allergies. Allergies are caused by our immune system's overactivity towards a substance that is generally viewed as harmless. We call these hypersensitivities because our immune system is overactive and shouldn't necessarily be responding in the way that it is. There are 4 basic types of hypersensitivity reactions, and we're going to discuss a few of them here now. Immediate hypersensitivities are also referred to as type 1 reactions or anaphylactic reactions. These are called immediately because they begin very, very quickly and they don't last for very long. The effects of immediate hypersensitivities can either be localized to a particular area or also systemic if the allergens can make their way into the bloodstream. Some of the common symptoms of immediate hypersensitivities are the characteristic runny nose, watery eyes, and itchiness. Generally, allergens that cause immediate hypersensitivities affect only the area that they come in contact with. This indicates that if allergens are inhaled, this is going to affect the smooth muscle of the lungs. However, if they're ingested, this is going to lead to other conditions that will pertain to the digestive system. These are some examples of more localized immediate hypersensitivities. However, if the allergen is able to make its way into the blood, we do get a systemic response, which can be very, very dangerous. Bee stings, spider bites, and penicillin can all lead to systemic anaphylactic shock. Individuals that are allergic to these types of stimuli will generally have to carry an EpiPen around with them. An EpiPen contains epinephrine, and this will be used to enhance the sympathetic response should your body start going into anaphylactic shock. Cytotoxic hypersensitivities are also known as type 2 reactions. We typically see these types of reactions in the case of a mismatched blood transfusion. If you're an A individual, you have the A antigen and the B antibody. And if we start administering B-type blood, the newly administered B antigen is going to interact with your B antibody. This generally does have a slower onset, however it does last a bit longer than the immediate hypersensitivities do. The last type of hypersensitivity reactions that we're going to discuss are the type 4 or delayed hypersensitivities. As the name suggests, these have a much slower onset, but they can last up to days. Poison ivy and heavy metals are 2 examples of substances that can cause delayed hypersensitivities. Poison ivy oftentimes does not have its effect until days after the initial exposure. These can often be caused by haptens. So as we come to the end of our presentation on the immune system, it is fitting to discuss how the immune system changes with age. As we get older, our immune system wears out and it becomes less and less effective. Generally, the older we get, the more immunocompromised we become. We tend to find ourselves more likely to fall ill due to infections, and our immune system does not respond to vaccines as efficiently. As with all of our body systems, age does take a toll on the effects of that system, but we must remember that this is a normal part of aging and a normal part of life. So it doesn't take this slide for us to see how vital the immune system is to the rest of our 11 body systems. It is the immune system that protects the rest of the body from all different types of substances that are trying to cause us harm. We have many different mechanisms for protection, and as we should be used to by now, all of our body systems are going to help out the immune system in some way as well. To finish up this presentation, I have a couple example questions that I wanted to give you guys to aid in your studies. The first question states, breast milk provides an infant with antibodies helping to protect the infant. What type of immunity is this? So first and foremost, we need to decide whether or not we're looking at active immunity or passive immunity, and then we can decide if this is naturally acquired or artificially acquired. So we must remember that active humoral immunity is when our B cells are challenged to produce their own antibodies. And passive humoral immunity is when antibodies are given to the individual and the B cells are not challenged, and therefore that does not establish memory. In this example, the antibodies are coming from the mother, and the baby is not having to produce its own antibodies. So that counts out this option and this option. So we know we're looking at passive humoral immunity, and the only question left is whether or not this is naturally acquired or artificially acquired. Well, the passage of antibodies from the mother to the baby is a natural process. Artificial administration of antibodies would come in an immune serum. So the correct answer to this question would be natural passive immunity. Artificial passive immunity would include some type of immune serum, such as an antivenom for a snake bite. Our second question states, which of the following cells requires antigen presentation to become activated? So one thing we know for sure is that macrophages are antigen-presenting cells themselves. So this cell is not going to need any additional antigen presentation, as it is the antigen presenter itself. And if macrophages is not the right answer, then all the above cannot be the correct answer either. We saw with the humoral immunity that B cells were actually able to be stimulated by the microbes themselves. So that means that our correct answer to this question is T cells. T cells are the only types of cells that require antigen presentation in order to become active and facilitate the immune response. This will conclude our chapter about the lymphatic system and the immune system. If you have any questions at all about the material, please make sure that you reach out to me through Canvas. I will see you all in our next lecture presentation when we will start to discuss the respiratory system.